Today I'm going to represent expression of excellent gene in eukaryotic cells. So our main aim of the project is the cloning of gene. So let's begin by discussing the ambitious goal of the project. We aim to clone the excellent gene into a specific strain of bacteria. Why excellent gene? It encodes an enzyme known as xylanase, which is crucial in breaking down xylane, a key component of a plant cell wall. This enzyme is not just a biological curiosity, it has significant industrial applications. Xylanase plays a vital role in biofuel production, pulp and paper processing, and even textile manufacturing. Coming to another slide, which is our strategy. Now let's delve into our strategy for inserting the excellent gene into the PC DNA 3.1 vector. This vector is highly copied copy number plasmid equipped with strong promoter optimized for gene expressions. The process involves several meticulous steps. We start by amplifying the excellent gene using PCR, utilizing specific primers for precision. Next, the vector is carefully digested with restriction enzyme, creating compatible overhangs for the PCR amplified gene. The excellent gene, PCR product, and the digestive products are then ligated together using T4 DNA ligase. This ligated DNA is subsequently transformed into E. coli cells with transformers selected based on antibiotic resistance. Finally, we isolate and sequence the plasmid DNA from these E. coli cells to confirm the accurate insertion of excellent gene reaction. Then we performed another step, which is known as transformation of cell culture. Here, first we prepared the cell to be receptive to DNA in bacteria. For instance, cells are treated with calcium chloride, which makes the cell membrane more permeable. Then recombinant DNA and E. coli taken in the plastic cuvette, placed on ice for 30 minutes. For bacterial transformation, a crucial step is heat shock. This provides briefly exposing the cell to a high temperature, which creates a thermal imbalance across the cell membrane, encouraging the uptake of the DNA. Then, after heat shock, cells are placed in the nutrient-rich medium to recover and to express antibiotic-resistant gene, typically included in plasmid. Finally, selection are plated on agar medium containing antibiotics. Only those cells have successfully taken up expressed plasmid antibiotic resistant gene will survive and grow. Then we find the validation of excellent gene insertion. Either the gene is inserted or not. For this we perform the transfection of the cell. First we use a microfuge to separate the bacteria from the culture. Next we added lysis buffer to concentrated bacterial cells. Then we pass the lysate, which contain our recombinant DNA, through a spin. The DNA bound to the silicon matrix in the spin column was then eluted using an elution buffer. To prepare for gel electrophoresis, we mixed 10 microliter of loading dye with our DNA sample. We ran our DNA sample in a gel made with 200 microliter of TAE buffer. Finally, the moment of truth. After performing the electrophoresis, we observed distinct bands corresponding to 5 kb for the plasmid and 1 kb for our gene of interest. This result confirmed that we have successfully cloned our gene of interest, making a significant achievement in our project, as you can see in this picture. First, we microfuge, then light disease buffer was loaded and it was ran into a column, silicon column. Then we put the recombinant DNA bind to the spin column. Elution buffer was added to release recombinant DNA. And after performing the experiment, we have our cloned DNA. Next, coming to another slide. which is our final step. We begin by growing by both streptomyces lividans transformant, which have been genetically modified. Next, we introduce the prepared DNA into competent cells by using calcium chloride and phosphate buffer. Once the DNA is introduced, we focus on protein extraction. After recovery, we incubate the plates. Moving on to main objective, we set up an assay to measure xylanase activity. This enzymatic assay is digestive, designed to quantify the breakdown of xylane into simpler sugar, a processed catalyzed xylanase. Finally, we analyze the data obtained from the assay. Our goal is to compare the xylanase activity between the transformant and the wild type cell. The comparison will reveal the impact of our genetic modifications on xylanase production, providing valuable insight into the efficiency of our genetic engineering technique. So it was all from my side. Thank you so much.